all of the data is available through the Allen SDK. It's also hosted in some other locations, such as the Dandy repository, but really the easiest way to get up and running is to install the Allen SDK in your Python environment, or to use this AWS environment that we set up. And then there are commands that make it really simple to browse through the data and get access to NWB files, and you don't have to worry about kind of where the files are stored and like manage the downloads yourself. So yeah, everything, the interface for all the data is this EC-EFIS project cache object. We use this term EC-EFIS, which stands for extracellular electrophysiology, because it's potentially more general than just Neuropixels data, but in this case, we've collected all the data with Neuropixels probes, so we kind of use those two terms interchangeably. The first step is to do the necessary imports, so these are already filled in, and then you have to decide where you want the data to be stored. So if you're running this on AWS, there is this path that's already predefined where all the data lives. I'm actually running this locally, so I have a path that points to a location on my local computer. One thing to be aware of here is that the files are quite large, so the NWB files are around 2 gigabytes just for the spiking data, and then if you want to download the LFP data, that's like sometimes as much as 7 gigabytes per session. So you want to make sure that if you're going to be accessing a large fraction of this data set, that you have ample space available on your local machine. But if you're running this on AWS, you don't have to worry about it. All that data is already on the cloud. So first step is to create this cache object that points to our manifest path. And then this will provide access to a number of files. And so these, similar to the 2P data set, there are these functions like get channels, get units, get probes that give you these give you access to kind of the metadata about all of the sessions and probes that we recorded from. So if we just want to see what sessions we have available, we can say cache get session table, and then so this gets loaded directly as a pandas data frame, and this is quite convenient because there's a lot of nice functions in pandas for filtering the data frame and kind of honing in on the data that you're interested in. And so yeah, if you're not familiar with the pandas library, I highly recommend doing some tutorials and learning more about that. We use pandas a lot in the Neuropixels portion of the Allen SDK. And you also saw some examples from Saskia. It's a very useful library, so I highly recommend learning that. So this is just the first 5 entries in this table. We can see the information that's available, such as the specimen ID. This is the ID of the mouse that we recorded from, which type of session it was. So I mentioned that there were 2 different stimulus sets that were shown. One is called Brain Observatory 1.1, and the other is called functional connectivity. So you'll see about half the sessions with one of those 2 session types. You can see the age of the mouse at the time of the experiment, its genotype. So in our case, we're using transgenic mice to do optotagging. So we have some mice that express channel rhodopsin in specific interneuron subpopulations, and then at the end of the experiment, we shine blue light on the cortex, and we can activate those cells and see which ones express the gene of interest. But we also record all the activity of the unlabeled cells. So this is kind of like an extra layer of cell type information that we get on top of the cells that we already record. You can get the number of units that were recorded for that session, as well as the number of probes and the structures that were recorded. So if there's a specific area that you're interested in, you can use this table to hone in on the experiments that have recordings from that area. But let's move on to the well, yeah. So for filtering these data frames, there's a kind of a standard syntax for filtering pandas data frames. You basically select the columns that you're interested in, and then say the values within those columns that you want, and then just use the ampersand to get the intersection of all these things. So this would give us a smaller table that contains only male mice with that express channel rhodopsin and somatostatin neurons, and use the Brain Observatory 1.1 stimulus, and include recordings from VisL, which is another name for LM, or lateral medial visual area. So this ended up with just 3 sessions, but there's still a ton of data in these 3 sessions. Yeah, that's just one example of how you might want to filter this data frame. So we also have a table that includes information about all the probes that were used. So each session includes data from up to 6 probes at once. So you can use this, get probes. So yeah, this is a table of all the probes that were used across all experiments. Some may actually be the same physical probe that's used in multiple experiments, but within each experiment, the individual probe gets a name, probe A, B, C, D, E, or F. And you can see the total number of units that were recorded by each probe, and which probes were in which areas. This is somewhat useful to pay attention to because typically the same probe targets the same area across each day. Kind of the probes are in a set configuration. So if you want to look at only the data from primary visual cortex, that's almost always from probe C. And so you could filter all the data that was recorded by probe C, and that will almost always include some cells from primary visual cortex. So I think that information is in the cheat sheet that's also available in the GitHub repository. Okay, next we can get information about the channels that were recorded. So yeah, this is within each probe. There are up to 384 channels. And so this is useful because we have, in almost all cases, we've mapped the channels to a particular location in the Allen Institute Common Coordinate Framework. And so you can say for each individual channel, not only what area is it in, but what precise location was it located in the brain. And so you can get this interesting spatial information and look at things like how receptive fields vary across different locations of the brain. See how things change based on depth in cortex or depth in hippocampus. So having information about the channel location is very useful. And then finally, we have our table of units. So this is the table that contains the metadata about all of the units that we recorded. So yeah, we start with the raw data sampled at 30 kilohertz. We extract units using kilosort, and then we apply some quality metrics to those units to kind of determine how well isolated they are, how much contamination they have, whether they're likely to be missing spikes. So I mean, one thing to be aware of is that there are varying levels of quality in these in every electrophysiology data set. And it's not so simple as dividing it into isolated single units and multi-unit activity. There's much more of a continuum here. So by default, we filter these units to only return ones that we would consider reasonable for most analyses. So we use these default filter values for ISI violations, amplitude cutoff, and presence ratio. There's a Jupyter notebook on the Allen SDK doc site that goes into much more detail about what these different what these quality metrics mean and why we've chosen these particular thresholds. But if you want to get access to every unit that is recorded, you can also do that by disabling these default filters. And so yeah, this will give us over 99,000 units. And so yeah, there's some analyses where you really just want to have access to all of the information about activity in a particular area. Like if you're doing a decoding analysis, you may not care if all of the cells are well isolated or not. But if you're doing an analysis of, say, bursting activity, and you really need to know exactly when every spike for a particular unit happened, and you want to be sure that you're not missing any spikes, then you want to be more conservative in your quality metric thresholds, and maybe even use ones that are a bit more stringent than the default values. Okay, so to wrap up the data access portion, I'm just going to make a pretty plot using the information in the units table for the spatial information for every unit that we've recorded. And so here, yeah, you can see we are sampling densely in cortex, hippocampus, and thalamus. The colors indicate the area that each of the cells is mapped to. So yeah, this gives you a sense of the scale of the data set, and hopefully gets you excited to dive into it a bit further. Okay, so those are kind of the tables that you can get that give you information about all experiments, but a lot of the analysis will require accessing data about spike times or local field potential, and those come from NWV files that are specific to one session. So if we go back to our the data frame that we pulled out of the sessions data frame, we can choose one of these sessions. So the index of that data frame is the session ID, and so in this case we're just pulling out the third index because the numbering starts at 0 in Python, and then loading this session using another function within this cache object called get session data. So that takes a session ID as input, and you can get that session ID by filtering the sessions data frame. Okay, so within each of these sessions, there is a units table. So this sometimes takes a second to read in all the information, but basically this will give you the metadata about all of the units that were recorded in this one session. And then that table will have as its index unique ID for every unit. And then you can pull out the spike times, et cetera, for each of those units. And the unit IDs within the NWB files are the same as the unit IDs that we originally pulled out in this very long table of units. So if you find a unit in this larger table that looks like it could be interesting, you can then figure out what session it's in, and then load the data for that session, and then access the information for those units. Okay, this is taking a second. Okay, so here we go. So yeah, this has similar information that was in the original units table. Some, like for each unit, there's some information about its waveform. So if you're looking for example, fast spiking cells in cortex, you can use some of those waveform metrics to pull those out. It also has information about the structure that it's in. This is anterior pretectal nucleus in the midbrain, and then what probe it's on, etc. So we can filter this table in a similar way to the way we filter the sessions table. So here we're taking all the units in primary visual cortex and then setting a bit of a more stringent cutoff on ISI violations. So these would be considered units in primary visual cortex that have lower contamination. So if this results in 35 units from this experiment. And then we can get the unit ID by looking at the index of this table. So this column on the left is the unit ID. We'll just take the first unit from this table, and then we call session.spike times to load in those spike times. And so one thing you'll notice here is that it gives this warning that said there are some invalid times in this session. This is not true for most sessions, but there are a few sessions that do have invalid times. Basically, it meant that there's something that went wrong with the data acquisition, and we want you to be aware of that. So basically, like, because we have 6 probes acquiring data at once, sometimes there's a problem with one probe, and just because that there's a short period of time when that probe's data is invalid doesn't mean that we want to throw out the entire session, because there's tons of data there. But this is something that's important to be aware of. But in order to figure out what went wrong, you can just type session.invalid times, and in a second it should show what the issue was. So in this case, there were about 20 seconds where probe E went offline. In our case, since we're analyzing B1, it's probe C, so we don't have to worry about this. But in general, this is not something you have to worry about, but we do want you to be aware of it in case you see like a section with no data, with no spikes. That doesn't necessarily mean that the cortex went silent for that period. It could mean that there's just data missing for that interval. Okay, so another thing you may be interested in is the waveforms for particular units. So we can load those just by using session.mean waveforms. And then the Allen SDK also includes a function called plot mean waveforms that will plot those for you. It's very self-explanatory. So in a second, this should pop up. And so yeah, the waveforms are actually saved for all of the channels on the Neuropixels probe. So you can see kind of this 2-dimensional waveform with lots of information about how the spike is propagating through the brain. But in this case, we're just going to pick out the peaks channel, so you can see the overall shape of the waveform. Okay, well, there we go. Okay, so yeah, here you can see the unit IDs as different indicated with different colors. You can see a combination of these fast spiking cells with a narrow peak to trough interval, and others with a more regular spiking waveform shape. Okay, so I will talk about how to access the stimulus presentation times, and then I think we'll probably move to Q&A. So similar to the 2P data, you can pull out the names of the stimuli. They do have specific names that you need to remember in order to filter the stimulus presentation table. That will come up in a second, but the using session.get stimulus table, you can get the stimulus table for a specific stimulus type, such as drifting gratings, and that will give you the information about all the presentations of that particular stimulus. Let's do one more section, which is the behavioral data. So you can also get the running speed for these sessions. It's just session.running speed, and that returns a table that has information about the start time at which the running speed interval was recorded, and then the velocity of the mouse at that time. And that will allow you to visualize and analyze the running speed. And there are definitely cells that are modulated by running speed. The responses in cortex can be quite different depending on whether the mouse is running or stationary. So that is something that is interesting to analyze. And then you can get information about the mouse's gaze location, both the size of the pupil as well as the location that was pointing.